Now we're going to do allopatric speciation, and it goes out to everybody who loves isolation or being isolated. So if we're going to start off with the original population has variation. Then we're going to state that the original population has gene flow. Now, gene flow within a population keeps the allele frequencies stable. Right here, allele frequency stable. Now, the allele frequencies in your course, you need to link that up with the idea of P and Q, and P plus Q must equal 1. So it might be, P might be 0.4 and Q might be 0.6, but it has to equal 1. P is normally the dominant allele and Q is normally the recessive allele. Okay? Then you can talk about sources of variation. You don't need to go into a great diatribe about what the variation is. You just need to state what causes it. And you should always mention mutation, and you can mention DNA replication if you want to. You can talk about, you can mention crossing over in prophase 1, and you can talk about independent assortment in metaphase 1 and metaphase 2, etc. You just need to mention that there's variation, and you need to mention that you know what the variation is caused by. Okay? So that's OP plus B. Then we get to this bracket in the middle, which is GI, and now we're going to be talking about a geographical barrier.
What is it, GI? Isolation.
A geographical barrier is going to separate or split the population in two. It doesn't have to be even 50-50 numbers. It has to just divide it in two. Right? And because different subpopulations are going to go to different sides of that geographical barrier, it's going to create geographical isolation. With the geographical isolation, you have prevented gene flow now. The individuals on each side can't access physically each other. So geographical isolation is preventing the two subpopulations from gene flow, from interbreeding. So the two gene pools now are isolated. Therefore, gene pool isolation. No gene flow. Whereas before, the gene flow was creating a stable allele frequency. By not allowing each population to access each other, it's not going to be stable anymore between the two groups. They're going to develop different changes. Okay? So then we've got DE. DE stands for different environment. And the environment is the combination of the biotic and the abiotic factors. When you're mentioning the geographical barrier, pick something sensible, hopefully to do with the question. If it shows you something that lives on land like a goat, pick a mountain range or a river. Now,
what can separate things in the ocean?
Land bridges, whether a river mouth opens or closes.
An island?
You're welcome.
Something. You just need to think. All right? Different environment, therefore you've got different combinations of biotic and abiotic factors. Right? Therefore, you're going to have different selection pressures. You'll need to say what a selection pressure is.
A problem posed by the environment.
A problem posed by the environment that differentially selects different phenotypes.
Okay.
Like, could you say that again?
I have recorded it for you.
Yeah,
but no but.
Same thing, really.
Okay. A selection pressure can only see phenotype of an individual, and it acts differently on different phenotypes. Okay, those with the phenotype that is an advantage is called a selective advantage. Those that don't have the correct mutation to get the advantages have a selective disadvantage. Yep. But the environment, the selection pressure, can only see the characteristic as it is expressed. It can't see genotype. It can only see what you look at, what characteristics you have, what enzymes you have, what morphology you have, what behavior you have, what biochemistry you have, what physiology you have. It can't see the genes. They are related, genotype and phenotype, but genotype is what genes you have, and phenotype is how they are expressed, how they look to the environment. Okay? So with different selection pressures, we're now going to have, due to the variation, remember we have variation, we have variation in the population, variation of phenotype. We have variation of phenotype. That's what the environment can see. That's what the selection pressures can see. And some of the variation will have the correct mutation. Therefore, some of it will have an incorrect or an unfavourable mutation. Those with the correct mutation, we say, has a selective advantage. They are able to solve the selection pressure. They can solve the selection pressure. Can't solve the selection pressure. Okay? If you can't solve the selection pressure, what's going to happen to you? You're going to die. If you're dead, can you breed? No. Are you going to pass your genes on to the next generation? No. That's the end of the line for your genes.
Why do humans not act like this?
So,
Because we have minds.
if you're able to solve the selection pressure, you're said to have a selective advantage.
Animals have minds too.
If you're alive, and therefore you'll live. If you're alive, can you breed? Yes. Breed, and the parents are going to pass their DNA or their mutations onto their offspring. The mutations are the correct mutation because they've been alive. I was trying to think of the right way to say it then. The parents, because they're alive, have the mutations, the good mutations that the environment has selected, and then they will pass those on to the next generation. This is one of P to F1 is one of the tenets of natural selection. Remember tenets of natural selection? Overpopulation, so more individuals are born than can survive.
Can we go
Yep.
over that, actually?
I'm going over it now. Heritable variation, which means the variation can be passed from, or the variation is mutations, and those mutations can be passed from the parents onto the offspring. Number 3 is competition for resources due to selection pressures. And number 4 is those with the selective advantage, or those with the correct adaptation, will survive to be able to pass on their genes to the next generation. Yep. But I can write them down separately for you if you would like. All right, I'll write them over here. Tenets of natural selection. 1 is overpopulation, which means that there is more offspring born than the carrying capacity. Good. Number 2 is heritable variation, which is the parents will pass on the mutations to their offspring. The variation is the mutation, the new alleles, the favourable alleles.
I didn't know you were
Right.
Number 3 is competition, which is different phenotypes compete for limited resources or compete for survival, but I prefer limited resources. But a resource can be escaping from predators, etc. All right, competition is between individuals with different phenotypes. Okay? And number 4 is survival of fittest, which means if you've got the selective advantage, you will live, you will breed, and you'll pass on your genes to the next generation. If you've don't have the correct mutation, you've got a selective disadvantage, you will die, and you won't breed, and you won't pass on your mutations to the next generation. All right? At about this point here, you can talk about genetic drift. genetic drift, which is a genetic bottleneck due to a natural disaster, or it could be the founder effect, where a small population breaks away. But a founder effect means you've got an isolated population, and now you've got those errors that we spoke about.
It's a sampling error that
The
causes
sampling
loss of
error,
an
which
allele.
means that not all individuals will breed, and not all of the alleles will be passed on to the next generation. There'll be the loss of alleles. So that's where we would now put in, that's where we would put in genetic drift.
Competition. Heritable variation.
Okay, that's the sampling error. Okay, the sampling error is the fact that not all individuals participate in breeding for the next generation, and also because you can only pass on half of your allele complement to the next generation, it's impossible for you to put all of your variation from both parents into the next generation. So you lose alleles. Once they're lost, they can't be reinvented. You need to wait for the mutation to occur again. So you lose the alleles from the population. Right? So now we talk about here, if the two populations were to physically come back together again, they would not be able to produce fertile and viable offspring. Viable means lived long enough to reproduce, and fertile means you can make gametes. Okay? So they might be able to physically complete the act of fertilization, but it won't end up in a life form that has the correct amount of genetic information to be viable in itself. And we call that reproductive isolation. Prezygotic isolation means barriers that stop two individuals from completing the act. Right? So it's pre-fertilization. So if we, for instance, square peg, round hole, the two sets of reproductive structures physically don't go together. Okay? They might have different mating techniques. They might have different breeding seasons. All of those things that are before the actual fertilization is one. It's called prezygotic. Postzygotic is to do with the genetic complement, like the mule. You know, where a gamete of 32 and a gamete of 31 come together to form a mule. But because now you don't have matching homologous chromosomes, you can't have a fertile offspring.